The final focus session this afternoon in the Centre for One Health annual conference. And this session is focused on people, nature and the environment. We're delighted to welcome a fantastic group of speakers this afternoon. So we will have, as with the other sessions, we will have our keynote speaker first, followed by 3 short talks given by early stage researchers. And I am delighted to welcome our speakers of this afternoon, Dr. Gertie van Keulen, Dr. Carol Bacon, Wengshuan Shui, and also Dr. Katrina Carlin and Michael Day, who will be our final speakers. So without further ado, I would actually like to start by inviting our speaker, Gertie van Keulen. And I just want to give maybe a little bit of background initially, just to introduce Gertie, but I'm delighted that she could join us. We would wish it was in person, but it's fantastic to welcome Dr. Gertie van Keulen from the University of Swansea. Gertie is an associate professor in microbial biochemistry in the Institute of Life Science at Swansea University. And she leads an interdisciplinary research and innovation teams with microbiology at its core. And the research that she's doing is basically expanding our understanding of and our ability to be able to sustainably exploit the chemical material properties of microbes in living, manufactured and in natural soil environments. And her research to date actually links antibiotics, understanding of antibiotics, water repellency and material. So that this enables insights from her work on microbial biochemistry through to manufactured soil systems and natural soil systems. And this enables step changes in knowledge and sustainable innovation in this field. So Gertie's talk today is on interdisciplinary advances in soil sciences, research and policy. And I'm delighted to welcome Gertie to our session.
Okay, so yeah, thank you, Maria, for the wonderful introduction. That's yeah, great to hear that you appreciate what I've tried to do so far. I am trying to go to, yeah, big screen is happening. Can I get a thumbs up if you can see my screen?
Yes.
Yeah, okay, brilliant. So yeah, so I've already Maria already gave you a title, and I will be able to go into some of our research that we've conducted in soil sciences area. But Maria and I also work together on policy work with the Microbiology Society, and I will touch upon that at the end of my talk. I'd like to give you a bit of a flavor of what my research and innovation interests are about. And as already indicated, capturing and exploiting the potential of my favorite bacterium, Streptomyces, as they are very prolific engineers of soils, of chemicals and materials, and a range of different research and innovation projects have arisen from that. I don't work on that full time on all of them. They go in and out of fashion also when funding arises. And yeah, you might think, what is that bulletproof glass with that bullet doing in here? So that's all linked to the work that we're doing on modulation, and particularly biomodulation of materials, whether they are microbial, whether they are natural, or whether they are manufactured, and insights from one are cross-applied to another. So our soil work that I will talk to you about today has also been used to develop materials for the defense sector. And part of that work has now also come back as interesting questions for our soil aggregation work. The key theme around the materials work that we do focuses on surface hydrophobicity, which, as you may know, is a physical property of a surface of material, where it seemingly repels a mass of water, which is indicated by water droplets here on a lawn of Streptomyces bacteria, water droplets on the surface of a water repellent soil, and a range of manufactured materials such as coated steel is shown here. So there's unlimited occurrence of surface hydrophobicity around the world. And you can say that there's a convergent evolution of sources and users, functionalities for the surface hydrophobicity. What that also means, and that's where I've been lucky enough to work at Swansea University, where there's a bottoms-up approach to interdisciplinary working. Approaches and methodologies from one discipline can easily be applied across to another discipline, and that's been instrumental into the advances that we've been able to make. If you've not heard of soil hydrophobicity before, or soil water repellency before, then that's okay, because many soil and hydrology textbooks actually don't mention it or don't refer much to it. It is the inability of soils to re-wet upon drying, and that manifests itself as beading of water on top of a soil, which you may have actually seen in like me, if you've got house plants but you're not too careful with watering them, so the soils dry out. If you then put water back on, then you see beading of water, and it takes a while then for the water to infiltrate in the top. It's a common feature across soil types, can be very fine soils to very crude soils, textures, different vegetation types, especially grasslands, are prone to development of soil hydrophobicity, but it's also occurring in many different climates. And what now are the consequences of that soil hydrophobicity? Well, if you're a house plant and you get your water, then you'll be fine and continue to grow. However, at natural soils or agricultural soils, what happens if soil hydrophobicity develops if you have reduced water infiltration, which then also leads to so-called preferential fingered flow, where water channels develop when it rains or when water is provided. However, there are large parts of the soils that remain bone dry, and that can cause then in turn reduced plant growth, because the plant root systems don't have access to water in an equal measure, and that can then reduce, for instance, crop yields, or in this case, very reduced patchy growth. If you're a golf fan, then particularly links courses are prone to water repellency in the soils. When the soil is water repellent, then you can also have enormous surface runoffs when water is then applied, when it rains. And that can also lead to higher pollution rates of agrochemicals and that type of thing, but also soil erosion, as you can see here. So this is my other half, it's almost 2 meters. So this is an indication of what kind of soil erosion can occur from a water repellent soil receiving water. COP26 is happening, so I've also looked at CO2, and my colleague Emilia Urbanek in geography has looked at soil respiration, and she found that soil respiration is affected by soil hydrophobicity indirectly because of different soil moisture distributions. The soil hydrophobicity is a transient phenomenon. Soils will re-wet after prolonged rainfall, and also when temperature drops. For instance, in winter, soils become water wettable again. If you then look at drivers and effects, with a population increase, then there is a greater demand for fresh water, more agricultural activity, but also more disposal of sewage or sludges, which leads to increased soil carbon content, which in turn can lead to increases in soil water repellency. Climate change itself may also affect soil hydrophobicity, because there are more drought periods, temperature increases, which in turn increases water demand and reduces the moisture levels in the soils. So you have these drivers that affect each other and can accelerate the development of soil hydrophobicity. What now then are the molecular sources of water repellency development? And that's been a long-standing interest, obviously, of the soil repellency research area. So molecular sources that have been implicated are small compounds, low molecular weight compounds, such as lipids and waxes, which are extruded, released from plants and other materials. Extracellular polymeric substances and mucilages have also been implicated, but also wildfire products release, for instance, charred products into the soil, which can increase the soil water repellency. And particularly in root systems, it's the mucilage that have been implicated there as well. However, if you look at these compounds, wettable as well as repellent soils contain equivalent amounts of these low molecular weight hydrophobic compounds. It's not just the presence of these compounds, it's also the orientation and the deposition nature that needs to be in the exact way to then develop a water repellent surface. What we focused on in this project is microbial proteins. And I'd like to introduce you a bit also going back to my postdoc work back in the University of Groningen, where we looked at amyloid proteins. And there are microbial proteins that have a critical role in microbial differentiation, in morphological development, but also, for instance, have key roles in biofilm formation and colony development. So these proteins are extruded by the microbes to remodel their own cellular surfaces, so that, for instance, they can enable to grow and survive in air, because these proteins produce a water repellent surface on one side, but a hydrophilic surface on the inside. And that, for instance, helps with prevention of desiccation of the cellular material. The extrusion of the material also enables remodeling of the external environment, of whatever nature that is. And that, for instance, helps with adhesion to materials, but also with colony development. And here are different pictures of these proteins. These proteins form amyloids, amyloid fibers, as shown here, in between the hyphal structures of Streptomyces. And as you can see here with atomic force microscopy on the top surface of an aerial hyphae. These proteins have converged as a result of convergent evolution, and they don't share any amino acid sequence similarities. So fungi produced on Streptomyces, bacilli, E. coli, all sorts of bacteria produce these amyloid proteins, and they have a functional role in these sources. But it's only the protein fold, the amyloid fold, that they share. There's no sequence similarity, which makes it, from a bioinformatics approach, more difficult system to approach. So what we then put is that what's the impact of these functional amyloid proteins on natural matter? So the project hypotheses that we worked under was that changes in soil protein abundance and localization drive the transitions between hydrophobic and hydrophilic conditions at soil particle surfaces. And these transitions, in conjunction with soil structural properties, strongly then influence hydrological behavior at the macro scale. So we used a wide range of approaches, covering from the molecular scale to core scale. And we've used proteomic approaches, atomic force approaches, goniometry at different scales, but also core scale wettability and water retention as more soil science hydrology approaches, and the network modeling of the void of the pore structure has also been applied. I won't be able to go into detail into all of these approaches because that could be an entire session in itself. So if you're interested in a particular approach, do let me know, and happy to talk after the session about these. And also because we wanted to do so many different approaches at these soils, we were not able to study a lot of different soils. So the soils that we were able to study were bulk soils collected from 3 locations across England and Wales. And the first one is Parkgrass experiment, which is a control site, but also a site that has many metadata sets, which, with respect to soil repellency, develops only subcritical repellency, so a very minute amount of repellency. And then we have 2 sites in Wales, close to Swansea, where very close to Swansea University, extreme repellent soil is present on Gower Peninsula. We sampled the soils under wettable conditions and repellent conditions, and then applied all these different methodological approaches to them. I will only be able to give a snapshot of a few of these, so focusing first on our metaproteomic approaches. We have worked on the total metaproteome as well, where we have done some methodological and bioinformatic advances to enable us to ID proteins better from soil metagenomes that have been deposited. However, I will only cover our amyloid extraction work, which we've termed the ultra metaproteome. To extract amyloid proteins, it's more of an enrichment protocol because you apply high temperature and detergent conditions to remove soluble proteins. So you end up only with the amyloid proteins that are very resilient to a range of environmental conditions. And if we extracted our different soil samples, we found that only the repellent soils we could detect protein, and we were not able to detect any protein in our wettable soils. However, if we spiked in, for instance, Streptomyces amyloid protein, then we could extract that from the wettable soils. So that suggests that these wettable soils do not have these amyloid proteins at the levels that the repellent soils have. Due to technological difficulties for amyloid proteins, you normally use Multitoph mass spectrometry to identify them because they tend to be poor subjects for tryptic digests. However, due to presence of humic acids and other brown substances, that caused a big matrix effect, which rendered the technique not very useful. So we resorted back to tryptic digests of these amyloid proteins, which is not the best way to study these because they often don't have tryptic cut sites in these proteins. So we have under-detected the amyloid proteins present in our samples. However, we were able to identify 19 proteins, and the sequences were present in our metagenomes. And if we run them through amyloid prediction software, then most of these proteins were returned as with an amyloid feature. If you then start to look at the biological origin, some of them were prokaryotic and some were eukaryotic, but most of them could not be linked to a particular known genome sequence of any organism. So we still don't know what the organisms are that produce these amyloid peptides in these soils. As you can see in this picture, there is one protein, M4442495, that is very abundant and were detected in all the 3 repellent soils. So we were very happy with that identification. And we studied that protein a bit further. So we ordered this as a synthetic protein. And we also discovered that it's not only can be dissolved in water, but it can also be dissolved in other solvents. And that makes it a protein of interest for our materials engineering work, where sometimes water is not a suitable solvent, for instance, bare steels. So in this case, if you have a hydrophilic substrate, glass, and a hydrophobic substrate, PTFE, or Teflon, maybe you know that better, then when you put a very thin coating of 10 nanometers down using this synthetic soil protein, we were able to modulate the water contact angle of these substrates. So here you see the water repellent nature of uncoated Teflon and glass as a wettable substrate. And you can see from the pictures taken here the different changes, which is quite dramatic. If you look at Teflon, the reduction of the water contact angle is quite steep, whereas the increase in the water contact angle upon coating of hydrophilic substrates is not as large as that we had hoped for, but we may actually not have the whole protein, or we may not have the right nucleation parameters that would produce a hydrophobic surface in soils. We do think, though, that with this protein, we might have the start of an environmental biomarker for bulk soil water repellency. However, we will need to validate this biomarker further by testing other repellents, soils, other soil types, different climates, etc. So if that's something that is of interest to you, then do get in touch. So we've only studied one protein, so there are 7 other candidates that together with the abundant protein might become a suite of environmental biomarkers for soil water repellents. So we were very happy with the results that repellent soils have these ultra proteins and wettable soils do not indicate the presence of these proteins. So we were quite keen to look at the results of the atomic force microscopy studies. So there's a lot of methodological development that has taken place to be able to do this, because AFM usually requires flat surfaces, whereas a soil particle soil is not a flat material. The postdoc who developed this work, Andrea Gazze, has been extremely successful and in diverting us away from potential mishap with where we first looked at features that we could explain in soil mineral context, but were actually quite the artifacts. So after resolving that issue, Andrea looked at first imaging of the soils, and the soil particles display all sorts of features, from flat features to the mineral stages to globular features to features that we were particularly interested in these fibrillar structures. And if you zoom in on this, these look cables of fibril structures. And of course, there are all different types of fibrillar material in surfaces present. But we were pleased to see that there is some resemblance to amyloid proteins when they are deposited on mica, which is shown in this picture. So physiologically, so just by image, topographically, the features are there that we hoped would be there. And with atomic force microscopy, you can also study different mechanical properties that are not possible if you use, for instance, scanning electron microscopy. And elasticity, etc., is in a similar range as for these features as what we've seen for our amyloid pure proteins. What we did not expect, though, was the result when we looked at these mechanical properties, and mechanical properties such as stiffness, deformation, adhesion, which is a measure of wettability, and in phase, which is a measure of the chemical signature of the material as such. And what Andrea found is that if you look at the nanoscale and compare wettable and repellent soils, is that there's no difference, no significant difference in adhesion. There's no difference in wettability. So that was a quite surprising result, because we had such a positive result from our ultra proteome. We were able to detect differences in deformation and in the chemical signature properties. So we were disappointed with that result, But then when the results from the water retention and the soil pore structure modeling became known, then it all started to fall into place. And that's what I'd like to introduce you to now. So this is work done by soil physics and hydrology modelers. And what is shown here is a soil pore structure where the voids, so the pores, are shown in brown. And what is not shown in this picture is the actual physical materials, such as the particles, root materials, other type of things that you might find in a soil. But it's just the voids and the connections between the voids that are shown. And here is a unit cell that's been built up using the water retention curve data for the different soils that we sampled. So this is an experimentally derived model, and the scale bar here is just over half a millimeter. So it's a significant pore structure. What you can do is not just rebuild the pore structure with Pore Expert. What you can also do is start to model, simulate water infiltration. And that is what's shown here at the top. So the water droplets start to move in from the top of the surface. And when the water infiltrates into the unit cell, it turns blue. And you can model that in a different way. When that was done, using the fully wettable void structure from our intermediate repellents soil, but on a wettable state, this blue line here indicates that water over time, as the more water you put on the top, then the water content is able to increase, so water infiltrates. And then different parts of the void model were closed off, and water continues to infiltrate if you remove the smallest features. So 5 microns and smaller, 10 microns and smaller are shown here. But we saw quite striking results when the features 30 microns and smaller were removed, where you have a hard stop here. At this point, water does not infiltrate anymore, and as a result, water will bead on the top. If you remove larger features, then the water infiltration stops even quicker. And to show you what that looks like, so this is a close-up of the unit cell, and in red you can see what the features are that are 30 microns and smaller that have been removed from this void model structure. So the larger pores are still there, and this animation then zooms in on how small these features are that are removed, that then result in such a massive change in water infiltration. So this modeling then shows that by limiting water excess to only 0.5% of these interconnecting threads and pore structures, you get a reduction in wettability of 70%. What the Pore Expert model also indicated was a good fit with another hydrology soil physics model of Van Verneuchten, which I won't go into today. So that started to explain and bring together both the proteomics with the nanoscale features that were not significant. Because if you look at then the process of repellency development, so soils dry out, and when you have a critical threshold level, which is different for each soil as measured by soil moisture, then soil water repellency can develop. As a result of reduced access to water, there is biological adaptation to that, and also increases in osmotic stress, which we can observe with total metaproteomic results, which I've not been able to show you today. But as a result of more air, more gas becoming available in these structures, these soil amyloid proteins are deposited, which probably reside then in micropore hotspot zones, because the AFM work indicates that there's very few of these surfaces that are water repellent, but also that it's not different between wettable and repellent surfaces. This heterogeneity was also apparent from our goniometry work, where you can determine water repellency at a nanometer to a micrometer scale, which can, with the water retention and the void modeling, has then helped explain these features in that if you need only to reduce 0.5% of the whole of the pore structure, which then already results in a quite dramatic increase in water repellency and water beading on the surface. The conclusion from this work is that you need to put interdisciplinary researchers together to really make great step movements into understanding of how soil processes work, and in this case, how water repellency works, and also indicating how at the different orders of scale of magnitude these processes do link together. And using modeling, we've then started to understand how these effects can be upscaled into higher magnitudes of size. As a microbiologist, I am pleased to say that the microbial activity at small scale can have major effects. So microbial activity and differentiation do matter as such. So this work was not done by myself, but by a great team of interdisciplinary researchers, as indicated here. So Andrea, Ingrid, Kat, and Jerry were the research assistant postdocs in this project. Which brings me then to our policy work that on the basis of our interdisciplinary soil water repellency work, I was invited to contribute to, which is part of the microbiology society's policy development work towards a sustainable future. And that was part of its 75th anniversary project to look at how microbiology can help and raise the profile of the United Nations Sustainable Development Goals. And this is also where I worked together with Maria, who took charge for one part of the policy development. So if you look at the microbiology component parts involved in sustainable development goals, they're grouped into food security, health, and environment, where I was involved with the development of soil health processes. There was also antimicrobial resistance. So these topics were picked before the COVID pandemic because emerging infectious diseases was a major theme in that process, but AMR was picked in this case. And biomass and circular economy is also of great importance to achieving the Sustainable Development Goals. So the Society held 3 online themed workshops on these topics. And I think due to COVID pandemic and people working from home, it was actually possible to bring more people together than was actually possible in person, what the original plan was. So looking at soil health, soil health has got, the functions are dependent on soil forming factors as indicated here. However, no 2 soils are the same, and therefore it's very difficult to pinpoint what a soil health, what a healthy soil is, because there's not a specific metric defined for that. So different administrations across the globe have come up with conceptual definitions of soil health. Of the one used by USDA is often used and cited. Framing then the role of microbes in soil health in the context of climate change, I don't have to tell this audience that climate change affects soils chemically, physically, and biologically. And there's a role for microbes in stabilizing soil structure, but also mitigating soil erosion and flooding, but also could be exacerbating these effects. So understanding the role of microbes in these processes is quite important. So same as for carbon storage and the processes listed here. One major problem that in the discussions, in the workshop discussions came forward is effective changes in soil microbial diversity and soil health remain unclear, partly also due because of microbial redundancy, resilience and resistance, but also lack of metrics. So what is the healthy soils microbial community? What should that look like? Soil health impinges on different parts of sustainable future, and I've taken a figure from the really excellent review by Lehman et al. On soil health. So if you've not looked at that yet, I can recommend that fantastic review. And here I've bolted onto these the different sustainable development goals that we chose as relevant for soil health development, policy development. The workshops were divided into 4 different themes, where a full morning was able to discuss different parts of these. Participants were drawn from across academia in the UK and Republic of Ireland, from research institutes, but also from industry, for instance, agrochemical industry, farming, but also social, regulatory and political institutions, for instance, those involved with the soil, with the water and air directives, whereas there is a lack of a soil directive. So each of the workshops chose 2 topics to select, and together that framed then the different recommendations and aspects discussed. I won't be able to go into them all together now, so I will only present to you the 7 recommendations that concluded the Soil Health Policy Report, which is able for free download from the Microbiology Society page. And I need to move this bit out of the side so I can actually see the top part. So recommendation 1 was that sustained research is required to enable to establish the role of microbiology in soil health, but also to link it to the different sustainable development goals. It's important that the concept of soil health is understood and adopted by all the stakeholders, but also that the metrics that metrics are developed, that metrics that work for all the different disciplines that work together towards soil health, from agronomy to farmers to scientists. So each should contribute to making to developing metrics that are measurable, but also have a meaning in the actual field and that don't cost an arm and a leg. There's also different recommendations related to training, because interdisciplinary working is at the heart of improving soil health understanding and achieving that. But the training of workers, of the workforce, is required, and communication between the workers, but also explaining that to the users is very much important. So the recommendations are there. It's too much to go read through them. But it was a pleasure to work on this and a pleasure to be invited. So I'd like to move on. My slides. So the acknowledgements for the policy work for soil health are indicated here. And I'll stop here. Thank you for your attention.
Thank you very much, Gertie, for a fascinating talk, and thank you so much for your time with us today. I have lots of questions, and what I would encourage for our people to do, our attendees, is actually to put your questions in the question and answer box, and we will actually take some questions for Gertie now at the moment, and we will have, after our 3 next speakers, we will actually have further question session at the end of the 3 speakers. So I suppose while people are getting their questions organized, I have so many, but I'm going to be careful. I'll only start with one. And I suppose, Gertie, I was really fascinated with the work that you've been doing on the amyloidogenic proteins, because again, I suppose we're so used to these proteins being, well, they're synonymous with diseases like Alzheimer's diseases in humans, and to find that they're actually present in the microbial community, and also to see that they play such an important role in water repellency is quite fascinating. And I suppose my question is, because the rhizosphere is such a comprehensive network, and there's lots of different organisms interacting with each other, have you any insights that there may be factors that stimulate the production of these proteins, or that can control the production of these proteins? So are there any insights from your work in that regard?
So most work on the understanding of the expression of these proteins come from individual groups who study, for instance, biofilm development or differentiation, so sporulation in Streptomyces and mushroom formation, for instance, in the fungal world. So there are, for each individual organism, there are a range of environmental cues that convalesce and then ultimately result in the production of these proteins, which is often seen as some sort of terminal structure. Whereas in a natural environment such as soil, it's a continuously changing environment. So the how that is controlled is not readily studied. But so the regulators, the transcriptional regulators, and the signals that they respond to for each individual organism is studied. But putting that together in a multi-organism setting, that's been not well studied. And it's also not known if each of these individual proteins, if you put them together, what then happens with the water repellency. But a very interesting question, yes.
Yeah, thank you very much, Gertie. And I've actually just received a question here that's actually linked in, probably very topical, because it's relating, I suppose, to what's happening at the moment in terms of the COP26 discussions. And the question is, what do you see the impact of climate change will be on, I suppose, considerations for soil repellency and moisture retention, but also maybe saturation of soils into the future? And how will this affect maybe global issues such as crop production and food supply? Big question.
Yes, as indicated, so there's it's not just the amyloid proteins identified. There are also, so for instance, wildfire has major implications on development of soil water repellency as well. And with climate change, more and longer dry periods, that will also result in more wildfire occurring. When there was pre-COVID, we had a hot summer in Wales, and my soil science colleagues traveled around Wales to sample soils, and they did not find in Wales that there was additional repellency developing, rather than, so you only need a few days of dry weather. You don't need an actual dry period, a drought, to develop water repellency. So in that respect, it's droughts would not result much in change in that respect, but the level of moisture in the soil will affect, for instance, soil carbon sequestration and microbial respiration, because it's only in the soils that have partial moisture where you have soil respiration, even in repellent soils. So if you have a drought period, then release of carbon into the atmosphere is actually quite low. So it requires careful consideration. And if you do a CO2 modeling from a release from the terrestrial environment, it's these heterogeneities are very difficult to capture in the models. And that is where advances need to be made in how to capture the heterogeneity across different soil systems across the globe into a more accurate CO2 release from soils.
And I think actually, Gertie, this comes back to your point about interdisciplinarity being really important, because I think all of us are working on our little pet microorganisms, and frequently we forget that there's a bigger picture that and that collaboration is so essential for the work we do. Gertie, I have one final question from somebody here who's asked, and it's a big question. It's on, what do you see as the greatest barriers to translation of your research into sustainable processes?
I would think disengagement from end users, I think personally is important. A lot of farmers, a lot of land managers are interested and keen to promote sustainable development. But the long-term nature that scientists require to actually have a better handle of what's happening, that is off-putting to users who rather use something that has an immediate effect on their practices. And the long-term nature of changes in soil systems, the patience required to, and the funding required to be able to make those observations, that which practices work, which practices don't work. That's also a major hurdle, because funding bodies only look at 1, 2, 3 year projects and don't have 10-year projects in mind.
Thank you very much, Gertie. And once again, thank you so much for such an interesting talk and for being a wonderful keynote speaker for our session. Really good introduction to the session. We may have actually some more questions at the end for Gertie, so we will take those at the end of our session. So for our next speaker, I'm delighted to welcome my colleague, Dr. Karen Bacon. Karen is a plant ecologist and a paleoecologist here in the School of Natural Sciences at NUI Galway. And Karen completed her PhD in paleoecology in 2012 in University College Dublin. And she spent several years in the UK afterwards, including at Leeds University. And we were delighted when we managed to capture Karen and bring her and entice her to come to join us here in Galway in 2019. Her research interests span paleobotany to modern plant ecology, and also it incorporates quite a lot of interdisciplinary nature because it includes plant biology, geology, and chemistry. And Karen is especially interested in understanding plant extinction and how plants respond to environmental change. So it's very topical in terms of the current session. She's also very interested in taphonomy and the science of fossil preservation and understanding the quality of information that can actually be obtained from the fossil record. And her ecological research currently focuses on invasive species, plant chemistry, ecosystem services, and plant awareness. And the title of Karen's talk, which has me fascinated, is Plants in Crisis. So I'm delighted to welcome Karen.
Thanks very much, Maria. Hopefully everyone can hear me, and I am going to try to share my screen now. Come on. So hopefully everybody can see that slide now and can hear me. So first of all, I want to thank Maria for inviting me to speak today. It's great to have the opportunity to share some of the research that my lab has been doing in NUI Galway for the last 2 years. So the title of the talk is Plants in Crisis. But just before I get into that, what I want to do is just briefly explain what my lab here in Galway is doing. So it is everything, as Maria has just said, from paleobotany, paleoecology to modern ecology. And let me just move that. And I look at everything from directly looking at modern plants today to thinking about how these interact with human created ecosystems. So we've done a lot of work with stately homes and things like that, and we'll pick up on that at the end of the talk, and also on invasive species. And what I'm particularly interested in knowing is how these two extremes can inform each other. So how we can use the fossil records to tell us something about modern ecology, and then how we can use modern ecology to tell us about the fossil record as well. So you might want to know, what are these crises in plants then? So the first one is that I want to show you a series of pictures, and if we had more time, I might ask people to write in the comments how many they can identify, but you can just think about this yourself as we go through. So I imagine that pretty much everybody can identify at least 6 of these without even thinking about it. Okay, so these lovely animals, I'm pretty sure that most people know the majority, if not all, of these animals. Now, what happens when I put these images up? Do you recognize as many of these? Can you name as many of these species? And unless you're very interested in plants, you probably can't. You might be able to identify maybe 2 or 3 of them, which is what our research has shown. That's actually pretty good going. But compared to animals, people, generally speaking, don't engage with plants in the same way, even passively. So whether or not you're interested in the animals that I showed you, you probably know what they are, but you probably don't know what these plants are. And this is a phenomenon that is called plant blindness, or also lacking plant awareness. So it's just that people perceive plants as being in the background. They don't perceive them as being exciting or relevant or important. They're just also there. So sometimes I show students a picture of a very out of focus bumblebee on a plant and to ask them to tell me what's in the photograph, and they tell me they can't really see what the animal is. They forget about the plants altogether. So that is plant blindness, or lacking plant awareness. And we've studied this previously with in Leeds, when I was in Leeds, and what we found was, sorry, I'm just trying to see if I can move this away somewhere. Not really. But what we found is that when we looked at students in the University of Leeds, we asked them whether or not we gave them a series of pictures and asked them to identify them. We did the same with the staff, and we did the same with the public. And what you can see here is that actually the numbers that people can identify are, I think, quite shockingly low. It's really, you're talking about maybe 5 or 6 is actually pretty good for people to be able to identify. You might say, why is this a problem? And it's a problem because we look after what we understand often and what we care about. So plants are often at the bottom of the queue, essentially, for protection or for research funding, and for people being fully engaged with them, and yet they are fundamentally important to pretty much every aspect of our lives. So an example of this is that rosewood was accounted for over 35% of all seizures by the UN Office of Drugs and Crimes in World Wildlife's database. That's the highest proportion of all wildlife recorded as seized in the database. So plants are among the most trafficked of wildlife as well. And cacti and cycads are often at very high risk of illegal trade for horticulture and ornamental purposes as well. And we wonder then if this is feeding into technology. And it appears that it is. So one of my undergrads a couple of years ago looked at whether or not certain smartphone applications were better at identifying plants than animals, and they were much, much better at animals. And this work has moved forward to one of my Masters by Research students, who's pictured there at the top. And he's looked at 6 different plant apps that can identify things. So some of them are generalists like Google Lens, and some of them are specialists like PlantNet. But what we wanted to know is, how good are they at actually identifying plants? So we've been looking at it as flowers and leaves, and how well the different apps do at trying to identify. These are common herbaceous angiosperms. So these are plants that you would find very commonly. And what we have found is that some of them are quite good, regardless of whether you're showing them a picture of a flower or a leaf, and some of them are not as good, to be kind. And particularly one of the more popular apps is Seek. And we see here that it's really not doing terribly well on plants. So this idea of lacking plant awareness is feeding into the technologies that people are now using to kind of say, oh, this is definitely showing me what this plant is, but actually they're not really as good as they maybe are implying at doing this. The second crisis then is extinction, and this is tied in very much to the biodiversity crisis. So we know that throughout Earth history, we have seen various different periods of mass extinction. And there's things go extinct and originate in the fossil record all of the time. We know that plants have gone extinct over time as well. However, plants do not tend to suffer the same rapid mass extinction that we see recorded in the animal fossil record. So when we look at that today, what's the scale of modern biodiversity crisis in comparison? Today, we see that nearly 40% of our plants are considered at risk of extinction. That is absolutely huge, and it's not what we've seen in the fossil record in terms of how plants have responded to previous environmentally driven mass extinction events. So what we're seeing today is something that is probably quite different, particularly in terms of how plants are responding. An example here is from the recent State of the World's Trees that was published a couple of weeks ago. 30% of trees are threatened with extinction, and in Europe, 58% of our endemics are at risk of extinction. This is more than threatened mammals and birds. And again, the question is this different to what we see in the fossil record? So this is one of the things that my lab is trying to investigate in Galway. And one of the ways to do this is to look at leaf traits. So when we look at plant responses to environmental change in the fossil record, we can see changes in how the leaves look or how the leaves are constructed that relates to their environment. And this then has related to whether or not plants locally at a fossil site maybe go extinct or do okay throughout an environmental challenge. So my PhD student, pictured here, has been looking at leaf traits. And again, we've looked at the Irish flora here because the Irish flora has been fully assessed to determine how each species, to give it a value in terms of whether or not it's at risk of extinction. So he went through the full Irish flora, coded up everything in terms of leaf traits that we know we have seen some changes in the fossil record, and then plotted it all up together. So this looks rather complex, but basically it's showing you that all of the different colors that represent different levels of threat according to the International Union for the Conservation of Nature. So all of these represent a different threat level. And they're all over the place. So if, for example, we were finding that critically endangered plants in Ireland had a particular leaf trait, if they were all had shared a leaf trait, they would form a little cluster. But that's not what's happening. So this really indicates that the leaf traits are not related to the threat status of the plants. And we'll be looking at this soon in a fossil flora. So Harrison is just back from having visited Stockholm and the Swedish Museum of Natural History, and he's going to apply the same to a fossil flora that is very similar to today's modern Irish flora. So we'd be really interested to see what comes of that. We also, a master's student who's pictured there is just about to submit her hardbound thesis copy, so we're delighted, has looked at this for a very geographically widespread and evolutionarily long family, the conifers. And what Tegan found is essentially the same, that there's no particular pattern of leaf traits versus extinction risk in the modern plants. So we need to do a lot more work on this, obviously, but what it's showing at present is that right now we don't see a connection between leaf traits and risk status. And that strongly supports the hypothesis that what's happening today is different for plants than what we've seen in the fossil record. The third crisis is invasive species. So when we look at what is pushing plants towards being at risk of extinction, invasive plants are often one of the top pressures. There's obviously also things like deforestation and land use change, but invasive species is also very high up there. And these are pictures from near Kylemore Abbey in Connemara. And you can see the this is Rhododendron ponticum. So this is a hugely problematic invasive species in Ireland. And you can see the woodland there is almost entirely dominated by it. Lots of people quite like rhododendron because they think it's pretty. And it is pretty, but it's also really problematic. When we look in under the canopy, there's nothing there. There's a few mosses and maybe the odd fern, and that's it. So this is having a huge impact on our biodiversity, our native biodiversity. We don't see any oak saplings, for example. So invasive species can look pretty, but they do not provide a healthy environment for our native plants and animals, and they have a huge economic cost as well. So if we look at Ireland here, this is showing the impact of invasive species, and it's really, really quite high. So the EU Biodiversity Strategy for 2030 recognises that invasive species are a massive threat to European ecosystems, and the numbers that kind of get thrown around for how much invasive species cost are astronomical. So there's a recent estimate of 26 billion a year globally, and in Ireland it's over 202 million, but that is a really old figure, so that's probably a massive underestimate. So what we're particularly interested in is looking at ways of trying to deal with invasive species, but trying to do it in a way that is good for the environment, so not by constantly using lots and lots of different pesticides, herbicides and chemicals. And we'll have a student starting on this in the next couple of months to look at this in relation to Japanese knotweed. And we're going to have a new student starting to look and work with Kylemore Abbey to look at rhododendron as well. So we've lots of exciting things hopefully going on there. But these are among the crises that plants are facing at the moment. So people don't recognize plants or their critical importance in our lives. They're not aware of the risks that plants face in terms of the biodiversity crisis, and it appears that plants are more at risk of extinction now than at any other previous mass extinction in Earth history. And we're basically just not doing enough to try and reduce the impact of invasive species as well. The figure that I'm showing you here shows you the contribution to biomass that plants make, and plants are by far the biggest contributors to biomass on the planet. We need to do more to protect them, and we need to do more to understand them, because plants are very clearly in crisis, and that means that we are too. So I want to thank you for listening, and just thank our collaborators and funders as well for this research. So thank you very much.
Karen, thank you for a wonderful talk. Really fascinating. We've lots of questions coming in, so I'm looking forward actually to talking to you about some of those questions that have come in. Some very nice comments as well about your talk at the end of the session. So in the interest, I suppose, of time and everything, what I'm going to do first of all is I'm going to invite our next speaker. And our next speaker is a PhD student, Wenxuan Shi, and I hope I'm pronouncing it correctly. Wenxuan is actually working as a PhD student in a very important area of research in agronomic evaluation of fertilizers, nutrient recycling and sustainable agriculture. And she's an early stage researcher in a European training network, ReFlow, which is based on phosphorus recovery for fertilizers from dairy processing waste. And Wenxuan is actually engaged in a PhD. It's a collaboration between Teagasc and our colleague here, Professor Mark Healy in NUI Galway. So Wenxuan, I'm delighted to welcome you and looking forward to hearing your talk.
Yeah, hello. Hello, everyone. My name is Wenxuan, and I am a PhD student and an early stage researcher working in the Teagasc and NUIG. Now I'm studying an EU Mercury project, which is ReFlow. It is about the phosphorus recovery from dairy processing waste. So today I will talk about the potential environmental and human health impact from the land application of dairy processing waste. Here are the contents I will introduce today, including the current problems in the food system, the opportunities and to solve these problems, and the introduction of dairy processing waste, the possible solutions to solve the risk from them, and what future studies would be focused on. Due to the COVID-19 pandemic, we found that our food systems can't be resilient to such crises as they are not sustainable enough yet. So, and our current food system can account for nearly 1/3 of greenhouse gas emission and consume large amounts of natural resources, resulting about the diversity loss and the negative health impacts, which is due to both under or the over nutrition. Therefore, there are many policies and strategies in EU to accelerate our food system to a sustainable one. Like the EU Green Deal and the farm to fork strategies. This is an important one for the agricultural part. And for these policies, we hope that we can transfer to a sustainable society and our past linear economy to a circular one. These changes can help us to have a more positive environmental impact and ensure the food security, nutrition, and public health. Like in the agriculture, these are the main practices from the policies. We will encourage the organic farming, optimize soil fertility and fertilization to avoid the excessive use of fertilizers, the sustainable use of pesticides, and improve the animal welfare. So the dairy processing waste is a kind of waste from the dairy industry. Since it is the largest food sectors in EU, so every day large volumes of dairy wastewater generated. And this kind of wastewater has high BOD, COD, chlorides, and sulfates contents and will cause the eutrophication after discharge. Also about 2.7 to 4% global greenhouse gas emissions from the dairy industry. However, the dairy wastewater has high nutrients concentration, especially in phosphorus, and low heavy metal concentration. So, which means it's a good feedstock for the nutrients recovery. Here is how this kind of waste generated and to be used. The wastewater from the factory will enter into the wastewater treatment. And after that, the wastewater will be discharged, and the sludge will and generate large amounts of sludge, which they are rich. Normally, the nutrients will concentrate in the sludge, and since they are categorized as biosolids, so they will be applied into the agriculture as fertilizers. So here is the characterization results from my study. Basically I tested the nutrients and the heavy metal concentration. We can see they are rich in the nutrients, especially nitrogen and phosphorus, and the heavy metal concentration. I compared it with the EU limits of biosolids. The heavy metals are quite low in it. However, there are still some potential risks from the dairy waste, as the studies about in it is not enough. So I assume that there are some emergent contaminants not tested yet in the dairy waste, and the pathogens in these kind of raw materials will probably cause the disease and the negative effects on the microbial activity after land spreading. And also we should focus on to avoid the excessive fertilization about this kind of waste. And in our project, we have 2 review papers published here, and if you are interested in it, in the possible risks, you can go to check it. And there are some solutions. The possible solutions to avoid these risks. One is to produce the secondary raw material-based fertilizing products from them. There are mainly 3 products can be produced, including the struvites from precipitation, and biochar and the sludge ash from the high temperature conversion. These 3 products can be referred as dubious, and this treatment can reduce the volume of dairy waste, which can solve the storage problem of raw waste and avoid the negative effects from the pathogens, and the nutrients will concentrate into these new products. Like some of them, like struvites, can be a more valuable fertilizing products to the market. And I also characterization all the dubious products, like no doubt the nutrients are concentrated in these kind of products, but the nitrogen will be lost since in the biochar and ash due to the high temperature. And the struvites is a very good potential fertilizer because they have very low heavy metal contents. And but in the biochar and ash, the metals would concentrated in it, and they are higher than their feedstocks. I also compared the heavy metal concentration with the EU fertilizing products regulation, because these products are expected to go to the market and as a fertilizer, not waste. So, so the heavy metals are lower than the upper limit. Another solution we are working on is to develop a calculation tool about how to spreading the dairy waste. Since these products are highly variable in about their nutrients and metal profiles, we need large work to characterize them at first, and then to calculate and optimize the application rates to avoid excessive fertilization and the metal accumulated in the soil. So this tool will be based on both nutrients and metal concentration. This will make sure they are applied in a safe way. And I have a paper published about the product's characterization and this calculation tool here. Here is for how the calculator looks like. It's now based on the Excel program. I set the maximum legal heavy metal additions into the soil from the EU, which can help us to see, based on the heavy metal contents, how many dairy waste can be applied. And then here is the calculation rates from their nutrients concentration. So at last, both of the calculated application rates will be compared, and the optimized one will be chosen. And in the future, we will study more about the emerging contaminants currently don't know in their waste to prevent the risk from them. And then we hope to improve this calculator through the fertilizer efficiency of their waste. This will help us to more to avoid the excessive fertilization, and also will to predict the metals accumulated into the soil. And thank you, that's all.
Thank you very much, Wenchuan, for a very interesting talk. And we're looking forward to questions because I know that they're coming in. Just some questions later after our last speaker has presented. Now, our last speaker is actually, and Wenchuan, if you could maybe stop sharing your screen there for a moment, that would be super.
Sorry,
yeah, no, you're all right. That's absolutely fine. Understanding. Our last presenters are Dr. Katrina Carlin and just Wenchuan, I'm just going to say maybe if you can.
Fine. I'm looking for the Zoom tab. I'll close it at first because I cannot come back.
No, that's fine. Thank you very much. Thank you very much. Our last presenters are Dr. Katrina Carlin and Michael Day. And both of our last presenters are actually on fieldwork today, and unfortunately internet connections are a little bit unreliable. So they've asked that we would actually present a presentation, and they're looking forward and be happy to take questions afterwards as well. And I just want to maybe give you a little bit of, and give both of them a little bit of an introduction. Michael Day is a second-year PhD student in the Applied Ecology unit here in NUI Galway, and he's investigating insect-based agricultural measures for conservation of grey partridge in Ireland. And he's really passionate about biodiversity and sustainability. And I've worked a lot with Michael on the CUSP project here and sustainable development goals that are part of our NUI Galway charter here. And I can vouch he's extremely interested and very active in this whole area. And Katrina Carlin is a lecturer in the School of Natural Sciences, and she's based in the Applied Ecology unit. And her research interests are biodiversity, but also very much about people's connections with nature and how this promotes pro-conservation behaviour. She leads the Nature and Ecosystems subgroup in NUI Galway's Community University Sustainability Partnership, which I've just mentioned a few moments ago. And this has involved, Katrina has been instrumental in leading sort of the development and creating the NUI Galway's Biodiversity Trail, which is really, it's really fantastic on campus. She embeds her research within 2 postgraduate courses, which are the Masters in Sustainable Resource Management, Policy and Practice in collaboration with the University of Limerick, and also the Masters in Sustainable Environments. And Katrina and Michael's talk is NUI Galway Biodiversity Action Plan and Near Health. So I'm just going to try and share my screen here with you, and hopefully this will work. And we will just begin the presentation. So I'm just going to, I hope everybody can hear as well.
Yes. Maria, maybe you can increase the volume a bit.
other groups as butterflies, butterflies, and even wasps, which also provide pesticides and are very important. Another action, conservation from butterflies, black thorn. Brown hair streaks have a very restricted function, only appearing around the burn, south Galway, and rare. These are the eggs which were discovered on campus by the training January 2020, and since then it's been much better to preserve make sure that this population doesn't die out by 2020. Of course, wetlands are very important to the planet, being that we're on the curve, and while we do have lots of this wetlands, one of the actions of the action plan is to figure out exactly what the first bit of plants haven't had.
Sorry, everybody. I'm aware that I'm just after seeing the messages now that actually you cannot hear. So apologies about that. I will actually start start everything again. Apologies about that. The recording, the actual volume on the recording itself is quite low, so this might be part of the reason. So I will just I will try to increase the volume.
Maria, when you share, can you tick the box that says take sound from the video? Because that should improve it, because then you don't have to do it.
Okay, I'm just going to try and see. Sorry about that. I'm just going to try and see if I can do that.
So when you're in the share screen, tick the box share sound.
Okay, just one moment. I'll just try that. So, oh, share sound. Thank you, Gertie. Sorry, I hadn't clicked that. Apologies for everybody.
In these wetlands are.
So I'm going to go back
so that we can
to Michael's presentation.
another.
So apologies about that. I will go back to the start of the presentation again.
The action plan consists of 11 actions that the college can take in the next few years to maintain and enhance biodiversity on campus. This is particularly important since NUIG borders the Corrib Special Area of Conservation. It was developed as part of NUIG's commitment to the UN's Sustainable Development Goals, which are 17 goals set by the UN to achieve for a better world by 2030. 5 of these goals tie in with the action plan, those being life above land, life below water, climate action, sustainable cities and communities, and good health and well-being. You may have seen these signs around campus, which indicate an area that's been enhanced for wildlife, and this is in partnership with the All-Ireland Pollinator Plan. Pollinator enhancement is not just important for bees, but also for other groups such as butterflies, hoverflies, and even wasps, which also provide some pest control and are a very important group of pollinators, particularly at this time of year. Another action is the conservation of brown hairstreak butterflies and the blackthorn that they use as their food plant. Brown hairstreak have a very restricted range in Ireland, only appearing around the Burren South Galway, and in North Tipperary. These are the eggs which were discovered on campus by Katrina in January 2019, and since then there's been much effort to conserve them, along with buildings and estates, to make sure that this population doesn't die out despite the campus being developed. Of course, wetlands are very important to the campus, seeing as we're on the Corrib, and while we do have lots of ditches and wetlands, one of the actions in the action plan is to figure out exactly what the diversity of plants and animals we have in these wetlands are, so that we can better conserve them in the future. Another action is the composting of disposable paper cups, which has only become more relevant with COVID and the increase in one-use products. This is being undertaken by Maria Tuhi's lab, where they take these 95% cellulose, 5% plastic cups, put them through a bioreactor using fungal enzymes to break it down into a digestate that can hopefully be used as a building material for something else. This offsets some of the carbon cost of the plastic that goes into these cups. The use of green infrastructure ties in with the UN's Sustainable Cities and Communities goal, with NUIG being committed to using green infrastructure such as green roofs, seen here on the engineering building, and living walls to increase carbon capture and offset some of the carbon cost of creating these buildings, thus leading to more sustainable environments. Now Katrina will talk about near health and how biodiversity can positively impact physical and mental health.
Thank you, Michael. Good health and well-being is about complete physical, mental and social well-being. And I'm now going to present to you some of the research in this area carried out by myself and my colleagues, Dr. Geisha Kinderman and Dr. Easkey Britton. Good health and well-being is dependent on a healthy environment. This has been recognised by the EPA and the HSC, who funded our research along with other projects. As part of the Near Health project, we wanted to know how people thought about nature, what their values were. And what they told us was that nature should be accessible to everyone, but that it wasn't. And they strongly identified how being out in nature made them happy, and not being able to go outside affected their mood and their well-being. And being outside also benefited their physical health and well-being. Once we knew what people's values were, we were able to put together a range of activities in nature. Some of these were in urban and rural areas, some of them included green and blue spaces, and some of them were of varying duration. I'm going to give you a snapshot from one that we carried out here on NUI Galway. Walking is one of the most accessible activities. We collaborated with CRE, the National Institute for Preventative Cardiology, to implement this activity. Participants significantly increased their step rate over the course of the programme, bonded with each other, and developed a keen interest in nature. Partnerships involving walking groups, local heritage and community organisations could easily implement this activity in their area. We collated our findings in the Near Health Toolkit, which you'll be able to find on the EPA website. It's all about connecting to nature to benefit your health and well-being, but in doing so, it also connects you to opportunities to take care of nature and to do more for conservation. We hope that we're going to be able to build on these here on NUI Galway campus. And I'm going to leave you now with some of the nature sounds that you might hear on NUI Galway's campus.
And thanks very much to Katrina and Michael there for a really lovely presentation and a fantastic end to our session today on plants, nature, and the environment. And if Liam is okay, because I know my colleague Liam Morrison is going to start the concluding session, but I would like maybe first of all just to take a few questions, because I know that there are actually some questions that have come in here in the and also comments to the talks for this afternoon. So we've had 4 fantastic talks, and I would like to maybe field a few questions for our early stage researchers as well too. Just some comments, first of all, comment to Karen, very interesting point for the awareness of animals versus plants. I think I'd have a better chance with a wider range of plants. For example, more trees and crops would definitely improve my result. And then there's also a compliment here to Katrina and Michael. Thanks, Katrina and Michael. I absolutely agree regarding connecting to nature. Having worked for years somewhere with no windows and therefore no natural light, I appreciate time in nature so much more. So these are some lovely comments that we received to our presentations. And we've actually I've just a few questions here that I've actually received, and one here for Karen. And I suppose it's actually a really, really good question. It's Karen, what do you think the human impact, what do you think is, I suppose, the extent of the human impact that's on driving extinction? So do you think that's actually significant? Do you think it's maybe a bit more complicated?
I think that the human impact in terms of driving extinction is incredibly relevant, and I think that what we're trying to look at through several projects in the lab by looking to the fossil record is trying to say, is what's happening now more severe than what has happened in the past? And when we look back to the fossil records, there's obviously huge numbers of caveats with this. It's not a perfect record. Not everything is preserved and all of these kind of issues. But when we look back to the fossil record and we see these primarily, not all, but primarily climate-driven mass extinctions, we see certain patterns. And when we look at what's happening now, that's diverging from what we might expect, particularly in relation to plants. So are we seeing, we might be at the cusp of the first mass extinction for plants, and that seems to be being driven by direct human activity. So yeah, I think it's a huge component.
And Karen, I have a second question for you as well, and it's about disappearing plants. And the question is, are medieval written records of, for example, medicinal plant use also used as a resource to establish the rate of plant disappearance or availability in a region?
So the way that we know things have gone extinct, so plants that are listed as extinct, that will go back over written records. So it's usually back over the last kind of 500 years or so. I couldn't comment specifically on medieval records. I actually think this is a really, really interesting idea, and it's something I'd actually love to look into. But so basically it's like, if you have a written record of this species in an area and it's not there anymore, there's a huge amount of work actually goes into then eventually saying that that species is extinct. But all of those old records, as far as I'm aware, they feed into the kind of global scale definitions. But in terms of kind of say declines or very local areas or things like that, I don't really think that's been done on a very fine scale, but it would be really interesting to look at it. But those old records are how we know something was here 100, 200 years ago, and it isn't anymore. So they are part of it, yeah.
And one final quick question is just in terms of what can we do to reduce extinction?
I think the best thing that people can do is to grow native plants. So if that maybe if you are lucky enough to have a garden, maybe letting a little bit of it just be what it is, just be wild. Don't mow it so often. I know people are probably well aware of the kind of no mow May thing, but that's actually a really good example of a lack of plant awareness as well, because that's entirely focused on pollinators. It doesn't focus on the plants fulfilling their life cycle, because you know, if you start mowing in June, we've not done anything for the plants because they haven't had time to finish their life cycle. And I think the other thing that people can do as well is try and source seeds that are at least regionally native. It's very hard to get native seeds for a lot of plants, but you know, it's possible. And if you don't have a garden, you know, have a window box, have pots, and it helps the pollinators as well, obviously, but help the plants too. And I think we have a beautiful native flora with really stunning plants. And I think if you leave your garden to just grow out a little bit itself, you'd be surprised at what might turn up. So that would be my advice.
Thanks a million, Karen. That's fantastic. I have actually got two very quick questions for Wenshuang as well. The first one of these is actually a question about sludge applications in terms of dairy waste. And just wondering how much understanding is there between the combined effect of metals at allowed and above threshold levels and emerging contaminants on soil health and ecosystem processes?
Sorry, can you repeat the question?
I can, yeah. So how much understanding is there of the combined effect of metals at both allowed and above threshold levels and emerging soil contaminants on, I suppose, ultimately on soil health and ecosystem processes?
Like about the heavy metals, there are the EU limits about it. It's mainly about the biosolids because it's a sludge, so it's kind of biosolids. So there are the EU have the limit on how many biosolids can apply into the soil, and there are also the EU limits about how many heavy metals can be applied into the soil annually. But the emerging contaminants, the limits is not a lot, mainly about some very common contaminants like PAH, these things. But there are we would say there are many types of emerging contaminants like the microplastics, and the other things from the industry progress, they are not listed by the law.
Okay, so there's still a lot of gaps to be met. Yeah. And I suppose I have one final question, and it's probably to link all of our speakers together in a sense, because I mean from the work that Wenshuang has actually been doing, we know that, for example, dairy wastewater contains lots of hydrophobic molecules as well. And these are going to affect soil hydrophilicity or hydrophobicity. And this links in with what Gertie is, I suppose, looking at in terms of the microbes in the soil environment, but also the impact could be significant on plant health as well and plant biodiversity. So I suppose maybe just as a final wrap up for our session this afternoon, maybe would any of our speakers like to comment on maybe, I suppose, that connectivity and the impacts, and maybe what we can think to what we might think we could do to address some of those considerations, or maybe to identify what's happening?
Well, about this part, like, to be honest, my project is more about the chemical characterization. So about the microbiological part, I'm not sure a lot, but of course, since the sludge is the raw material, so definitely they will have the like the effect on the microbiological activities. So since we don't know how will they affect the plants, so as I talked in the we will do the fertilizer efficiency, use the real pot and the field trial to see how the effects and if the different types of sludge will have the good or a positive or negative impact on the plant's growth. Yes, this is what we will do.
Thank you very much, Wenchuan. Yes, Gertie? Yeah, good.
I would like to add, particularly also what came out of the policy workshops, is that a lot of people do fantastic work, very interesting work, but we're unaware of each other's work. So we need to come up with ways to effectively communicate with each other across disciplines, but also find ways that, because your projects, PhD projects, postdoc projects are usually set on these is the project plan. I think funders should also be better aware and allow for a more like a wild card establishment of a project strand, so that aspects that have not been thought of before, that only become apparent while the project starts, should be introduced. And that should also be communicated more effectively to politicians, stakeholders, etc. It's not just one defined part, it's also affecting and impacting on other research areas.
Thank you very much, Gertie. And once again, I would like to thank all of our speakers for this afternoon's session. And I realise I'm actually running out of time as well, because we've had a very interesting discussion. So thanks to everybody. Thanks to Gertie, thanks to Karen, thanks to Wenchuan and to Katrina and Michael for a really interesting session this afternoon. So now I would like to hand over to my colleague, Dr. Liam Morrison, who will close the conference.
Thanks, Maria, and I promise I won't hold you except a minute. I suppose I should begin today by thanking the panelists, all the presenters, all the participants. Thank you for taking your time to join us, and also a special thanks to all the keynote speakers today and to the early stage researchers for their presentation. And thanks to our three chairs as well. And I guess a final thanks to our Wright Institute comms team, Mohan, and to our support team, Siobhan and Niamh, who made it all possible. And also a special thanks to the director for our Centre for One Health here in NUIG, Dervla. Over both days, I think we have all learned a lot from each other that should help us in our research within the area of One Health. I know I certainly learned a lot today. And I just want to finish off by bringing to your attention the next event in our Spotlight series, which is entitled The Control of Parasites in Livestock on Farms. And that is hosted by Professor Dalton's group here in NUI Galway, the Molecular Parasitology group. And that's taking place on November 25th next from 11 to 12:30. And you can follow us and the centre on @OneHealth_NUIG. So thank you very much and enjoy the rest of your day. Thank you.